All right, please. Please be seated.
Hereby, I open this academic ceremony in which Anxhela Habibi will defend the academic thesis, Protease-Activated Receptors in Vascular Smooth Muscle Cells, a bridge between coagulation and vascular remodeling. My name is Harry Schout. I'm the pro-rector today, and I wish to invite you to give a short presentation of your thesis.
Dear pro-rector, dear members of the Corona, dear family, friends, colleagues, and for those following us online, in the next 15 minutes, I will present the main findings of my PhD thesis entitled Protease-Activated Receptors in Vascular Smooth Muscle Cells, a bridge between coagulation and vascular remodeling. Per chi parla italiano, ci sarà una traduzione sotto ogni slide. I would like to start by introducing you to coagulation. Coagulation is the body's emergency repair system. When a blood vessel is damaged, the coagulation factors are activated to eventually form a clot and stop the bleeding. Imagine your blood vessels as city streets. When the road is damaged, the construction crew rushes in to fix the road. Coagulation factors don't only form a clot. They can talk to vascular cells. When a coagulation factor is activated, it interacts with the vascular cell. And this interaction leads to shape changes, cell division, and cell motility, all important to repair the damaged vessel. What enables coagulation factors to communicate and trigger important changes in vascular cells are protease-activated receptors, or PARs. And vascular cells don't just have one type of PAR, but they have four: PAR 1, PAR 2, PAR 3, and PAR 4. Imagine PAR receptors as a doorbell that coagulation factors need to ring in order to talk to the cells. A variety of cell types express PARs, but a very important one are vascular smooth muscle cells. If we look inside the aorta, the biggest artery in our body, we see where VSMCs are located, surrounding the blood vessel. VSMCs have an important role in the vascular system. They control blood vessel diameter, therefore regulating blood pressure, and they also provide structural support to the vessels. Under healthy conditions, if the vessel is damaged, together with coagulation factors, we also find growth factor. Growth factors come in to tell the cells that they need to wake up and grow and repair the vessel. So together, these factors help maintain VSMC's balance in the vessel wall and help repair the vessel when needed. However, when signaling becomes excessive or dysregulated, as in the early stages of vascular disease, the same signals can push VSMCs to turn into synthetic cells. These VSMCs do not respond anymore to normal physiology. Therefore, I will refer to them as rebel VSMCs. When these rebel VSMCs accumulate, instead of protecting the vessel, they drive vascular remodeling. When blood vessels remodel, this makes it harder for blood to flow because of the buildup of an obstruction or plaque. This is called atherosclerosis. If for instance, a plaque ruptures, and this quickly forms a blood clot that completely obstructs the flow, we happen to have a heart attack. Heart attack is an acute event of cardiovascular disease, or CVD. CVD is the #1 killer in the world, with 20.5 million deaths every year. Although we know a lot about the risk factors for CVD, we still need to understand what happens at the cellular level inside the blood vessels to find better medications and treat CVD. The aims of this thesis are to study the role of PARs in driving the transformation of VSMCs into rebel cells responsible for vessel remodeling, and to identify new ways to block this process. To achieve this, we used 3 different approaches. In Chapter 3, we used human-derived stem cells, and we cut the PAR1 receptor from the DNA with CRISPR, a molecular scissor. And then we transformed these stem cells into VSMCs that completely lacked the PAR1 receptor. In Chapter 4, we used mice with a small cut in the DNA called mutation, where part 2 is. We then isolated the aorta and obtained VSMCs with a defective part 2. In Chapter 5, we studied what happens to the vasculature when we block the coagulation factors, so they don't talk to PARs anymore. We tested 2 different anticoagulants, warfarin and rivaroxaban, and then we took the aorta out to see the effects on vascular remodeling. In Chapter 3, we compared the effect of coagulation factor in VSMCs with and without PAR1. When we treated the normal VSMCs with the coagulation factor, we saw that the cells became rebel, releasing harmful signals, such as vesicle release, inflammatory signals, and cell stress. But when we treated with the coagulation factor our VSMCs that lacked PAR1, these harmful signals were not there anymore. They stopped. Next, we also studied the effects of the growth factor, since it is also responsible of making rebel VSMCs. So we saw that the growth factor is also able to generate the same harmful signals from VSMCs as the coagulation factor. But to our surprise, when we treated with the growth factor the VSMCs lacking PAR1, we saw that these harmful signals were once again stopped. These results, so based on these results, we suggest that the receptor of growth factor, or the doorbell of the growth factor called PDGF receptor beta, might be working together with PAR1 in order to drive harmful signals from VSMCs. Further studies are needed to understand how the PAR1 and PDGF receptor beta crosstalk happens, but we suggest that this could be a promising targeting strategy to halt vascular remodeling without disrupting the coagulation via PAR1. In Chapter 4, we studied the effect of the growth factor on VSMCs with a normal or defective part 2. The defective part 2 is present on the cell surface, but cannot be activated anymore by any coagulation factor. It's like having a broken doorbell. So when we treated with the growth factor the cells that still had a functional part 2, they released vesicles, and inside these vesicles we found small messengers. These messengers are known to be able to tell healthy VSMCs to turn into rebel ones. And once again, when we treated with the growth factor the defective part 2, we saw that less vesicles were released and less small messengers were present, meaning that these vesicles were less powerful to turn healthy VSMCs in rebel 1. These results suggest that part 2, together with the growth factor, push healthy VSMCs into rebel 1 via small messengers carried in vesicles. And blocking these small messengers could be another potential target to fight vascular remodeling. In Chapter 5, we investigated the effect of 2 different anticoagulant medication on vascular remodeling, warfarin and rivaroxaban. While warfarin blocks multiple coagulation factors, rivaroxaban is directly blocking coagulation factor number 10, which directly talks with part 2. Warfarin is a blood thinner that has been used for decades to stop dangerous blood clots to form in patients with atrial fibrillation, which is an irregular heartbeat, or patients with an artificial heart valve. Warfarin, however, causes also too much bleeding. That's why it needs constant blood tests to make sure that the dose is safe. Long-term use of warfarin has also been linked to calcification, which means bone-like structures form inside the arteries and makes them stiff, like heart pipes, reducing healthy blood flow. On the other hand, rivaroxaban is a newer type of blood thinner that also lowers the risk of clots, but is generally easier to manage because it doesn't usually need constant blood testing. To compare the effect of these two anticoagulants on vascular remodeling, mice were given either a high-fat diet alone that makes mice more prone to develop vascular disease, or the same diet with an addition of warfarin or rivaroxaban. But we also wanted to investigate the effect of switching anticoagulation therapy, so we first gave mice warfarin for 6 weeks and then switched to rivaroxaban for the remaining 12. We measured different parameters to assess the extent of vascular remodeling, such as the size of the obstruction, the risk of rupture, and the amount of calcification in the plaque. When we compared a high-fat diet with warfarin, we saw that, indeed, warfarin resulted in an increased calcification. When we compared warfarin with rivaroxaban, we saw that these features were decreased. And to our surprise, when we switched from warfarin to rivaroxaban, we saw that these features were even more decreased. Therefore, these results show how warfarin has the worst effect on the vessels, while rivaroxaban proved to be a better anticoagulant option with less vascular remodeling. The interesting finding is that switching from one anticoagulation therapy to the other results in even less damage to the vasculature. Therefore, these findings might be translated in the clinical practice to reduce vascular remodeling and keep the vessels healthy. As I come to the end of my talk, I would like to give you a few take-home messages. I hope that I convinced you that coagulation is more than just clotting. It talks to VSMCs via PARs. Understanding how PAR1 interacts with the growth factor receptor might help to develop new therapies to control vascular remodeling. Modulating the small messengers inside the vesicles released by VSMCs could be a novel way to prevent vascular remodeling. And switching from warfarin to rivaroxaban might be a better option to improve vascular health. So finally, my thesis shows how protease-activated receptors act as a bridge. On one side, coagulation is a vital repair system. Sometimes coagulation crosses the bridge in order to heal the vessel after injury. However, other times, the coagulation factors cross this same bridge to turn vascular smooth muscle cells, together with the growth factor, into rebel ones. So the challenge here is not to destroy the bridge, but to be able to regulate better the traffic across it and allowing repair without turning it into disease. Even though we already have treatments for cardiovascular disease, most of them manage the symptoms rather than stopping the disease at its roots. Therefore, we still do and we still need CVD research to find new medicines to prevent the damage before it happens. And with this, I would like to thank you for your attention, and I give the word back to the Prorector.
Thank you for this very clear presentation. The opposition will be opened by Professor Hacking, Professor of Biochemistry, Maastricht University.
Thank you, Mr. Prorector. Dear candidate, first I would like to compliment you with this really nice thesis with the beautiful metaphor of the bridge on the front, although I would be hesitant to step on it because it looks like a bungee type bridge. If you step on it, you go into the abyss and you'll be shot up to the moon, and otherwise it might engulf you and eat you. So it's a bit of a scary bridge, but I got the point with your last slides that it's really a relevant bridge. Your work has led to many new insights and corresponding papers, and of course these compliments to you also apply to your promotion team professors Schurgers, Hoef and Kauske. So with that, that's the compliments. But now we have to discuss the content of your work, and we would like to engage in a philosophical discussion about the word canonical. Because you start almost in the first chapter on page 18, you mentioned that coagulation factors have non-canonical functions as signaling molecules. But on page 107, you say canonical PAR cleavage by all proteases. So could you please make up your mind? What is it, canonical or non-canonical?
Highly esteemed opponent, thank you for your kind words and for your question. So what I meant at the beginning of my thesis introduction is that coagulation factors per se are not there only to form a clot and activate other coagulation factors in the cascade. They act also as signaling molecules. So in this case, I referred to non-canonical as in they do much more than just clotting.
Yeah, but then the better word would be pleiotropic.
Pleiotropic, exactly.
Good.
I think I agree with that.
Do we agree?
Yes.
Yeah. Because canonical, if you consider the work of your supervisor, Professor Roef, and also your opponent, Professor Mosnier, then I mean the PAR cleavages by coagulation enzymes would be canonical as very established as well, or?
Yeah. So at the end of the thesis, I referred to canonical and non-canonical to the type of cleavage of the PAR1 receptor that can be cleaved by thrombing in a canonical way, but can also be cleaved by other proteases, such as MMPs, that cleave at different sites of the receptor,
exactly.
Therefore eliciting different downstream signaling effects.
Yeah, but I think, canonical is being used nowadays as a very fashionable phrase to just indicate the first discovered activity of a protein. And this is something that we have to correct sometimes, because enzymes are basically autistic. They see this P3 to P3 prime, and they just cleave it, whatever it causes. So I think then we would agree on that. Okay. All right. So let's summarize your work in a nutshell. So PAR-mediated vascular remodeling governed by factor 10A. Would you agree? Say yes.
Yes.
So which protein would you think that jumps up in my mind now?
After you said factor 10, part 2.
No, factor 10A.
10A, tissue factor.
And? Go on.
Thrombin.
No, tissue factor, go on.
Pathway inhibitor, of
exactly.
course.
You know me so well. Okay, so I looked into your thesis, and TFPI was mentioned only once.
Okay.
And that was quite a disappointment. And even then, as an insult to injury to me, it was only mentioned with data not shown. So if you go to that page, that is page 86, you say, in your knockout cells, increased tissue factor levels are unlikely to be due to the loss of tissue factor pathway inhibitor, as DAG analysis, differential gene expression, I had to look that up, showed an increase of 2.5-fold gene expression in PAR1 KOs in TFPI compared to the wild type cells. And then, of course, this is also an easy one. If you then look at figure 5H, how then do you explain that the thrombin generation is higher in the knockout than in the control cells, if the knockouts have 2.5-fold higher TFPI levels?
Well, the DEGs only tell us something about the gene expression, and we don't really know about the activity of the TFPI itself.
That's very correct.
And they have a higher thrombin generation because PAR1 knockout, we showed that they had an increased expression of the protein tissue factor, and then they also had a higher activity. So they were more powerful in inducing thrombin generation.
Yeah, that's absolutely correct, because like in the previous figure, you showed that tissue factor goes up like 10 times. And even then, if TFPI would go up 2.5 times,
they would not
level up.
help it.
But I have to inform you that the the conditions under which you study the stromal generation are quite harsh because the cells make a lot of tissue factor. So you might have diluted your tissue factor. One of your paradigms should know and should have told you that if you want to study TFPI, you want to reduce the amount of coagulation trigger, so around 50 nanomotors of thrombin, because then you will pick up the tissue factor pathway inhibitor activity. But this is correct interpretation, and I'll still go with you. In one of your references, and this is reference 140 on page 62, you refer to a paper by your primer supervisor and our enlightened Professor Spronk, in which he describes how rivaroxaban promotes regression of advanced atherosclerosis in ApoE knockout mice by targeting coagulation factor 10A. With your results that you obtained with your research in this thesis, can you now explain why rivaroxaban will not only attenuate atherosclerosis, but could also regress atherosclerosis? Do you think that would be possible or feasible with your theories? It can hold atherosclerosis, I do agree, but can you find a reason why it should regress atherosclerosis?
So in our settings, we saw that, so we still have to check the 6 weeks of warfarin to be able to tell whether switching to rivaroxaban is regressing or is just preventing from the plaque becoming worse. So in our setting, we didn't try a regression to check for regression.
No, that's not about the observation. Could you explain why it would regress? Why it could?
I would think that probably by stopping the activation of the PAR2 receptors in the smooth muscle cells, this would lead to less phenotype switching, and they would probably start also sending themselves signals to keep themselves healthy. So we saw that some VSMCs also have the ability to send EVs with microRNAs that can indeed promote the contractile phenotype. Okay. So maybe it's that, but it's a speculation.
Thank you. It's a speculation, but I'm very happy with the answer, and I give the word back to the prorector.
Thank you. The opposition will be continued by Professor Schmid, Professor of Pediatrics and Adolescent Medicine, Heidelberg University. Thank you for joining via the internet.
Thank you very much for giving me the opportunity to read your thesis, and I really appreciated reading it and reviewing it. A lot of very interesting results, and I take the advantage now to learn a little bit more from you on this very interesting factors. I'm a pediatric nephrologist, so by nature I am interested in chronic kidney disease, and as you mentioned in your introduction, this is one of the important cardiovascular risk factors, and as you probably know, 10% of the population now suffer from chronic kidney disease. Could you a little bit elaborate on what the role of whatever chronic kidney disease is, you may describe this a little bit, may play in your described system of vascular disease?
Highly esteemed opponent, thank you for your kind words and for your question. So if I understood correctly, you asked about how my data relates to the vascular outcomes in CKD patients. Is that correct? Okay.
Exactly, yes.
So from what I know, in CKD patients, they have a lot of inflammation going on, and they have also increased oxidative stress, and they have increased uremic toxins present in their blood. And I think that this high inflammation and oxidative stress is also a stimuli for protease-activated receptors to increase their expression in smooth muscle cells. So I think that then once the microenvironment is not favorable anymore, such as during the high inflammatory states in the CKD patients, then these muscle cells might also then change and express more PARs, and these PARs get more activated, and therefore starts kind of a vicious cycle that then induces cells to be synthetic, to deposit calcium, and transform themselves into osteochondrogenic cells, and therefore leading to calcification. This is how I would interpret it.
Yeah, exactly. I think that's the relevant point. The problem, of course, is when you talk about chronic kidney disease, you have numerous different factors coming into play. But at the end, the question is, what does it in your system, in the PAR system? And this would be a very nice way to continue research from a perspective of a nephrologist. Then I would like to come a little bit back to the vascular calcification topic and talking about regression of vascular disease. Do you think and how regression could be achieved and what type of regression you could expect?
So what I would do is probably check the levels of APC protein, because it's known that APC-APCR signaling in endothelial cells and VSMCs, it's beneficial rather than detrimental. So in this case, we could promote then the signaling of PAR1 via EPCR receptor to be able to rescue the signaling in these cells. And then I would probably monitor this, the levels of DP-UCMGP in order to see how the levels of UCMGP are. And then I would probably supplement with vitamin K in order to keep their vitamin levels high in order to prevent a bit the calcification that's happening.
To prevent, but regression, I mean, let's be simplistic. With atherosclerosis and we have vascular calcification, I mean, what do you think the PAR system and vitamin K would act on?
So vitamin K would act on the MGP levels, and therefore having more carboxylated MGP, then the cells will be protected from calcification. On PAR, I think that probably by stopping the highly inflammatory environment around, then this could lead to downregulation of the receptors, and therefore they would probably tell the cells to become quiet again.
Okay, I think at the end it's speculative. I totally agree on that. And the most severe state of chronic kidney disease is dialysis. And you mentioned your data, which is more favorable and less favorable. And as a clinician, as a pediatric nephrologist, I would also, of course, be interested on the role of heparin and low molecular weight heparin. Do you have any data? And could you please explain a little bit the effect of these anticoagulation on PAR and the subsequent damage?
So in our research, we didn't do directly check for heparins or their effect on PARs, but I know that fondaparinux is a type of heparin that directly targets factor 10A. So maybe that could be an interesting drug to test to see if indeed by targeting 10A using heparins, the PAR2 signaling and inflammation also goes down. Yeah.
Yeah, okay, so there's hardly any data on that, I think, and I think that would be another way to continue because it's so frequently used in the clinical setting in patients with severe disease, and the cardiovascular risk increases by 40-fold in patients on dialysis, and we give them a lot of anticoagulation in clinical routine, so that would be another way to continue your very interesting research. And then I have a very easy question, maybe you don't know it, but it wouldn't be a problem. Any age dependency in PAR expression activity? Do you know anything about that? I'm just curious. Age dependency, you know, we are putting children on dialysis at the age of 25 weeks of gestation, so very young children, and we do dialysis in 90-year-old patients. And do you know whether the system is age dependent?
I am not aware of any differences in different expression in PARS in age and young, but I would guess that because adults are also under more risk factors than young patients, so smoking, high-fat diet, and they might be more prone to have bad vessels, so to have more inflammation probably in their vessels, and this could lead to have higher PAR receptors in the aged population compared to young patients.
Okay, thank you very much. Very interesting data, and I think many things you can, or we can study now based on your findings. Thank you very much.
Thank you. The opposition will be continued by Professor Mosnier, Professor of Translational Medicine at Scripps Research Institute in La Jolla, United States. Coming.
Thank you very much. Dear promovendus, first of all, I'd like to congratulate you on putting together a very interesting thesis. I have read it with a lot of joy and interest. One of the things close to my heart is PAR 1. And so I wanted to talk to you a little bit more about that, and especially about your ideas for translation of your findings. And I know this is, there's no data, you haven't studied it, but I'm very curious to hear your thoughts about that. In one of your propositions, your first one, you say targeting smooth muscle cell PARs to inhibit vascular remodeling may offer therapeutic potential, but it entails significant translational challenges. So of course we know that there is a clinical PAR1 antagonist on the market for Paxar. So let's start off, but could you explain a little bit the benefits or disadvantages or risks that come with using something like that's already on the clinic, like for Paxar?
Highly esteemed opponent, thank you for your kind words and for your question. So I know that Vorapaxar has been discontinued because it induced too much of an increase in bleeding. And I think that's also, it needs to be considered that when I say that it's challenging to translate to human because PAR1 is expressed by quite some cells in platelets, endothelial cells, smooth muscle cells. So it is very difficult to target coagulation only because of the variety of the cell types that express PAR1. So my approach would be with the PDGF receptor beta to first find out whether these two receptors actually physically interact because there's nothing known. So I would probably try to co-immunoprecipitate them or do a proximity ligation assay in order to check whether they're physically there. And if they're not physically interacting, then it's probably due to some downstream signaling effects that they both share. And therefore, I would investigate which, for instance, G protein receptor is activated by the PDGF receptor beta once it's phosphorylated. And then study how blocking this interaction ameliorates the vascular remodeling. But also in mice, it's difficult to study coagulation because they have part 3 and 4 in their platelets, not like human that have 1 and 4. So yeah, this might be a bit of a challenge because they don't have the same receptors.
Right, of course. So yes, there's challenges with inhibitors or drugs is that they have to be site-specific, and you mentioned that they're on platelets and other cell types, but also activity-specific. And so, you have you mentioned that a little bit before, you have the protective cleavage of APC, and then you have cleavage by thrombin, you have cleavage by 10A in this case. So it is a challenge to come up with something that you could use. So in your idea of what would an ideal compound look like? What would the characteristics of the ideal compound be? Regardless of if you can find it, but what would that entail?
I think the first thing that we should do is to make something that targets specifically VSMCs in order to stop them from gaining 9 or 10 different phenotypes in the atherosclerotic plaque. But it's difficult to target cell specificity. So probably the great thing would be to have nanoparticles delivering these small molecules that block the interaction or block the signaling of PAR1 and PDGF receptor together, and so that it happens only in smooth muscle cells.
Right.
So in mice, we could probably start a mouse cell line that it's selectively targeting PAR1 PDGF receptor interaction only in smooth muscle cells. And therefore, then you induce with ApoE background and high-fat diet atherosclerosis. And then you see how the VSMCs handled this by probably having less phenotype changes. This is what I would expect.
Right. And then a quick final follow-up. So we talked about PAR1 mostly, but you also have a lot of data on PAR2 that also interacts in that complex. So do you think that it's a complex of PAR1, PAR2, PGF beta receptor, or are they individual islands that do different things?
That's a good question. I think, so because we saw that when we delete PAR1 from the cell, PAR2 is also highly expressed. I believe that it might be in a complex. So it's known that PAR1 and PAR2 heterodimerize. So I think it's worth to also check how the PAR2 is interacting with the PDGF receptor beta, and if these three are all in the same group hanging out. So that would be easier then to target. If we know that more things are involved, then it will be more targeted, for sure.
All right. Thank you very much, Richard.
Thank you. The opposition will be continued with Professor Reinhardt, Professor of Clinical Biology, Thrombosis and Hemostasis at the Medical Center Johannes Gutenberg University in Mainz, Germany. Thank you for coming, for joining via the internet.
Thank you for giving me the word. So, Anxhela, so I was really excited, and it was really interesting to read your thesis, and it's a really very mechanistic and broad work. So I would like to, maybe one of the points that was most fascinating for me was the role of these extracellular vesicles as carriers of information. So could you maybe describe a little bit what those extracellular vesicles are good for and what kind of information they can transfer?
Highly esteemed opponent, thank you for your question. So we saw that when VSMCs, mouse VSMCs switch into the synthetic phenotype, they have increased vesicle release. And in these vesicles, the pattern of microRNAs expression was different than in contractile phenotype. And when we looked inside the sequence of these microRNAs and we matched them with their target gene of interest, which they are supposed to stop the translation for, then we saw that a lot of the microRNAs, there are some microRNAs in the synthetic VSMCs that were procalcifying, or at least they were targeting genes involved in procalcification, and they were also targeting genes that would promote proliferation, which are two important phenotypic outcomes of smooth muscle cells when they go into phenotypic switching. So we saw that these carriers have an impact on the transcription of genes, important genes involved in the phenotype switching of smooth muscle cells.
If you think of the size of those microvesicles, and I think there are also exosomes, so could you separate them out, and what were the methods how you dissect the exosomes from the microvesicles? And is there a difference in the generation of those vesicles? So could exosomes potentially also carry RNAs?
So we, in order to exclude any different contaminants, we ultracentrifuged the serum that we treated the cells with, and then collected the VSMCs, the EVs. We centrifuged again to get rid of the large particular bodies, and then when we analyzed them by size in the NTA, in a nanoparticle analyzer, Then we selected specifically the range between 30 and 150. So this would be the range of exosomes. But we did not really check for specific markers on these EVs to see if they were coming from microvesicles or released from blebbing by the membrane, or they were coming from the multivesicular bodies. That we didn't do, and I would have liked to do that, to have a more insight in the mechanism of how these microRNAs are loaded inside the EVs, for instance.
As a follow-up, can you explain a little bit how the PAR1 signal then is transduced for the generation of the microvesicles? I think you need at the end also a calcium signal. So how is that happening?
So I know that PAR1 activation and PAR2 lead to calcium signaling. I'm not aware of how they would directly influence the loading in the microvesicles, but I saw in a paper that factor 7 on PAR2 on endothelial cells was generating EVs that were very high in a specific microRNA. So, and they found out that there was a RNA binding protein that was responsible for this loading of the EVs. So probably I would go and look for RNA binding proteins that are probably modulating the pool of RNAs, and then see whether they are up or down regulated after stimulation of PAR1 or PAR2, for instance.
And the specificity of the signaling of PAR1. So is this PAR receptor really then specific for thrombin, or how do you have ways to dissect identify the relevant proteases that might be involved in your system? So what do you think about that?
Yeah, so we have mice that have specific point mutations in PAR1, so that R41Q means that it cannot be activated by thrombin anymore, but leaves the receptor open to other proteases, such as APC. So by using this point mutation specifically, then you could probably dissect whether it's really the PAR1 thrombin cleavage, or if there's some other proteases that can be involved.
Because this might be very context specific, depending on the environment of the cells, and also maybe on other cells that are activated or contribute with proteases. So thank you. Then I give back to the prorector.
Thank you for your contribution. The opposition will be continued by Dr. Proudfoot, visiting scientist at the Brabham Institute in Cambridge, United Kingdom. Thank you for coming to Maastricht.
Thank you. First of all, I wanted to say just how much I enjoyed reading your thesis. Incredible amount of work in there. And thanks so much for bringing it to life in your really, really clear presentation that you give today. Thank you. So first of all, I had some questions that just struck particular interest for me in chapter 3. So that's where you were using human primary cells as a model to investigate PAR1 and thrombin. And I just wanted to ask a little bit more about the cells that you used and where they came from and how many different isolates you picked for your experiments. Did you have many different human donors for those experiments?
Highly esteemed opponent, thank you so much for your kind words and your question. So in chapter 1, we used both primary human VSMC, so isolated from patients, and these were primary, so we could only keep them in culture for very few passages. And also, you're never really sure about the purity of these type of populations. Then to study the role of PAR1 and to be able to cleave the PAR1 out of the cells, so to make a knockout, we used stem cells. We isolated stem cells from the blood and then transformed and differentiated them into VSMCs. Unfortunately, we could only do this with one donor. So we had one donor, which was our isogenic control or wild type. And that's on the same donor we did the CRISPR-Cas to generate the PAR1 knockout. Ideally, we would have had at least 3 or 4 wild type stem cells and then had 3 or 4 different knockouts of PAR1. So this would be ideal. But in my case, it took us 2 years to generate the first PAR1 clone. So it was quite challenging to be able to have different donors. But also now with the techniques also advancing, it's much faster to obtain a knockout. So it would be definitely needed to check this thing with other donors.
Okay.
Sure.
And I was wondering also in the wild types, did you also sequence their F2R locus? Did you also look to check to see if they have any particular SNPs or polymorphisms that are known?
Yes, so in the wild type, when we sequenced, we didn't find anything specific as to polymorphisms that are associated with any disease. We did sequence the knockout, and we found that it was only one nucleotide insertion at the very beginning of the protein sequence. Therefore, this of the gene sequence. So therefore, this was disrupting the frame, and that's why how we could obtain a full knockout.
Okay. Another thing I thought was really fascinating was how you refer to these smooth muscle cells as rebel smooth muscle cells. And it did also make me wonder that if you, because I think you noted that the cells you used were, the primary cells anyway, were between passage 5 and 7. I wonder if you ever took it beyond that to senescence. And, well, whether you can tell me if you tried to do that or not, and if you didn't, I guess, what would you predict? Would they be even more of a rebel type cell?
Yeah, we
perhaps?
didn't really go super far with the passages, but I would think that going on with the senescence, they would start probably upregulating more PARs. This is what I would predict, because they would have more defects in their DNA. They would probably become more pro-inflammatory and releasing also vesicles that might be influencing as well their phenotype. So I would assume that, we would probably be able to find a lot of differences in the senescence ones.
Interesting. And just one last question about the cells. You used aortic cells throughout, even with the mouse cells you worked with. I was just wondering if PAR expression is any different in different vascular beds. You know, if we're talking about, I don't know if you have any evidence about coronary smooth muscle cells or smooth muscle cells from leg vessels that become particularly prone to atherosclerosis and calcification?
So we ourselves did not really check this because we were more focused on the aortic smooth muscle cells of the thoracic area. So also in the mice, I would always isolate the thoracic part, also because they have a different origin, embryological origin. So we would try to keep it as constant as possible, but I think there are differences that needs to be discovered and studied for sure in the coronaries or also in the abdominal aorta to see how the PAR1 expression is in the abdominal part where there's also aneurysms forming. So this could also
Interesting.
be. Yeah.
And if I may, just one last question that I was quite curious about. And so there have been some recent studies which have shown, like large population studies, where they've actually measured PAR1 in plasma or serum and found that if you have higher levels of PAR1 in your plasma or serum, that it's very much associated with some inflammatory diseases, with type 2 diabetes and some types of cancer. And it made me think, well, how is this receptor that's sitting on the plasma membrane, how does that get into the plasma? Is it some form of necrosis or extracellular vesicle release? Or another mechanism, and I just wondered if you might have had any clues about, you know, maybe did you ever look in conditioned medium from any of your cells in culture? Because that might help to kind of understand how these receptors could possibly leak out into the plasma.
I would say maybe maybe some platelets are also releasing some some little EVs from platelets. So maybe it's coming from there, but they could also come from the membrane probably of endothelial cells, because it's also expressed in endothelial cells. But indeed, it would be useful to know where is it coming from, and then use specific markers of specific cell types to check if in these vesicles we find markers from endothelial cells or markers from platelets, and then maybe we have a clue of where these EVs are coming from, if it's in EVs. I don't know where else we would find them.
Yeah, no, good answer. Thank you very much.
Thank you.
I'll finish there.
Thank you. The opposition will be continued by Professor Huge, Chancellor Professor of Vascular Biology and Vascular Biomedical Engineering at the University of California in Irvine. Thank you for coming to Maastricht.
Thank you. Dear candidate, I'd like to echo what everyone else has said. It's a pleasure to read. It's beautifully written. Your use of analogy was excellent. I think it really helped set the scene. I have several questions, some probably pretty short, some kind of more expansive. You talked a lot about PDGFR-beta. There's another PDGF receptor. Did you look at that at all? Did you think there's any involvement of that?
So, sorry.
It was PDGFR-B and PDGFR-A, two different receptors.
Hi, esteemed opponent. Thank you for your question. We did not look in the differences in the PDGF receptors, so we only looked in PDGF receptor beta expression, because we treated cells with PDGF-BB, which mainly signals through the receptor beta.
Okay.
That's why.
I was going to ask about mechanism, but you covered that already, so that was good. You're looking at the shift in phenotype of this cell, and obviously there's a natural process that is going to shift the cell back and forth as the body needs it. Pathological conditions, it seems to get stuck. Do you see that these treatments that you might be envisioning would have to be permanently pushing the cell back to where you want it, or could you just flip it and then it's going to stay?
So the dream would be that if you have a switch and then you press it and then they stay like this.
Yeah.
But I don't think that's feasible, because humans are different and they are also under different risk factors, as I mentioned before. So probably it's something they would need to keep taking to be able to keep it healthy, if they still want to be unhealthy on their private lives. Because I think already prevention and taking out the main risk factors would be beneficial. But I think,
So you sort of opened up a line of questioning there, and you said people are different. They're certainly different to each other, also immensely different to mice. So you did some work with human cells in culture, you did your work in vivo with the mice. Any differences between the systems in mice and human that you might be think might be significant for your work?
So
mice don't get atherosclerosis naturally, right?
Yeah, so it's difficult because mice are useful because you can study in vivo what happens in the plaque itself, and you can look at the different cell types generated and do some tracing and see where do these stems, VSMCs go. But humans I like to work with human IVSMCs because we could reach basically a pure population of VSMCs, and phenotypic-wise, they would also be a better model than mouse isolated primary VSMCs, which we were also constricted in the passage way.
Yeah. Okay. rivaroxaban work, I mean, you sort of suggested there that it is reversing the phenotype, right? And if I got this right, you said that it's almost better to give warfarin first, then rivaroxaban?
Yeah, so
What is going
it
on
looks
there?
like it. We were also very surprised to see that. So, but I was very careful not to use the word reverse in my thesis because we are still ongoing with analysis of the 6-week plaques. So we don't know whether warfarin mice have at 6 weeks a lower plaque than the high-fat diet, or if it's bigger. This we don't really know. So once we have to look into this 6 week and see if the plaque is bigger than the high-fat diet, then probably it is reversing. But if the plaque is smaller, it means that an initial warfarin treatment, so pushing down altogether the coagulation factors and coagulation cascade, might be beneficial initially to prevent some PAR-related activation of the cells. But then long treatment of warfarin, then you have the effects of the loss of MGP, and then you have the calcification taking over, and therefore we probably don't see this good effect of lowering the coagulation anymore. So we still don't know what's happening, but I'm looking forward to this result. And
so the calcification then you don't think is going to be happening long-term with rivaroxaban?
It's not, or?
Will it not happen?
No, because warfarin itself, in the way the warfarin works, stimulates calcium depositions, and of course of the loss of MGP, and the cells become more osteogenic. So it's, they are going to end up like that, basically, with warfarin. Whereas with rivaroxaban, we are not touching any MGP, and we're only focused on the part 2 signaling not being activated in the rivaroxaban group.
Very nice. Do I have time for one more question? Okay. I've been around a while, and I remember when Russell Ross first started talking about atherosclerosis being an inflammatory disease. And I think now everyone's pretty comfortable with that idea. When you've looked in your mice after the treatment with rivaroxaban, have you looked at macrophages? Do you see any change in the inflammatory component of the plaque?
No, we didn't, but I wish we had, because this would have also told us where the part 2 signaling would come from, because macrophages themselves also have part 2 expressed. So we would probably be able to see and discriminate if the part 2 signaling that we see of the expression is from VSMCs or from macrophages infiltrating. And this would give us more insight into how the plaque is formed and structured.
Right. I finally, because nobody stopped me yet, you've done an amazing amount of work, but as we all know, there's always something left to do. What's the one experiment you didn't do which maybe you wished you had done? Or if you had a chance to do it now, what would be that killer experiment you'd want to go back and do?
Generating the part 2 knockout from human stem cells.
Yeah.
That one we couldn't do, because the part 1 already took a lot of time to generate. But if we indeed could do, what I was planning to do was to generate a part 2 knockout induced VSMCs, and then on the part 1 also knockout also take away the part 2 in the same cell to see then the different effects of the loss of both receptors, or only one of them, to the VSMC. So this is something that I would like to do. And then the analysis of the 6 weeks animals indeed add inflammatory markers to see, maybe also analyze the vesicles from the mouse to see if they were having more or less tissue factor present on their surface.
Anxhela Habibi, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
All right, please. Please be seated.
Chair. Anxhela Habibi, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Schurgers is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right, please.
My second pair of eyes. So before we start, Anxhela, I have to ask you something. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Anxhela Habibi, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of our university.
Please be seated.
Now my bookwork starts. Dear Dr. Habibi, dear Anxhela, congratulazione. I'm the first one here to congratulate you with this fantastic achievement. And you did a really, really great job. And this is the highest academic degree that one can reach. So congratulations with that. I also want to take into these congratulations your supervisor, Professor Roof. Thank you, Wolfram, for taking this journey with us. And Dr. Croucher here online from Böhringer Bieberhaag. Thank you, Stefan. I also want to extend my congratulations to your partner, Kostas, and to your family, your friends, and your colleagues, and of course, your two paronyms. I wanted to start first thanking the members of the assessment committee. Professor Hacking, thank you for taking the role as chair. And also from Maastricht, Professor Youth from, well, actually Irvine, but you're also part of our Maastricht family. And thank you for traveling here to Maastricht and joining this ceremony. Professor Musnier, also from Scripps Research Institute, not university, all the way from San Diego. Thank you for coming here. Diane, thank you for traveling from Cambridge. It's a true pleasure to see an old friend here again. Thank you very, very much. And of course, also online, Professor Schmidt, welcome, all the way online from Athens. Well, actually in Greece, we've for the first time met, so it's really great that you're here. And also thank you very much, Professor Reinhardt from the University of Mainz. So thank you, we have a very international committee here for you present today. Dear Anxhela, 1 out of 15 ESRs, early stage researchers in the European ITN T-Cardio. Actually, and you were assigned ESR number 3. A European collaboration between Mainz, Maastricht, and Marseille. It's really a great group of friends that really brought something to the table. With the aim of generating a new type of young researcher that studies the interactions of the vessel wall with the blood, influencing cardiovascular disease, and that we term thromboinflammation. And your work really greatly contributed to that. It's a network of colleagues and friends, and actually the 15 ESRs were selected from all over the world. Greece, China, India, Germany, Italy, Chile, Spain, Kazakhstan, and finally Albania. Or is it Italy? Or the Netherlands?
Both.
So this was a very wonderful group of young people, and we also as supervisors enjoyed it really to have you as young people. Dear Anxhela, our story starts somewhere end of 2018, beginning 2019, when I was visiting together with Cengiz Bonn, where I have a long-standing collaboration with Catherine Cazzola, and she is working with me on the V course, and you were also doing the other work on V course C1L1. And we directly had a click, but back then we didn't really had a go on the grant yet. But when we received the grant in 2019, I directly contacted you, and our official interview was together with Dr. Elisabetta Castoldi, and my Italian colleague. And actually, we quickly offered you the job, and we were very happy that you took the job in Maastricht, because later I learned that also Bonn had offered you a PhD position. So we were really, really happy that your project was coming to Maastricht and Mainz, and where we had this double PhD with Professor Roof to work on thromboinflammation and the role of smooth muscle cells. The official start was the 1st of January 2020, and I guess most of us still have active memories around that time. It was when COVID, 1 month later, turned the world to a standstill, but also in the Netherlands, everything was closed down. Together with Vanessa, your paranymph, and also ESR number 7 in the T-Cardio Consortium, you started, and also she was actually selected from Bonn University. So you were there, well, locked down in a new environment, family at home in Italy, and actually together with Professor Hacking, we fought to keep our research lab open, and we succeeded. And I think we were, maybe even in the whole Netherlands, well, one of the few labs that continued, because we are working in a very safe environment. And so we tried to help you also to stay active and not locked down in your rooms. So we had to make the best out of it. Together with Vanessa, the girl sitting next to you in the office, you became more than colleagues, even true friends. And that was not based on a match in characters. You both were and are very different. But together you became a kind of Siamese research twin. Both of you working very, very hard, very motivated and very organized. And organizing many, many after-work get-togethers with the group. I can also only be very proud to see that very young people, next to working hard, also grow as persons. And that they have fun both in work and in life. And this enjoying of work even went to the next level when Aria, your other paranymph, joined our group. It seemed as if work was turning into a party every day. And I still remember the laughing sound out of room number 1334 that easily could reach my room and also Professor Hacking's rooms at the very, very end of the corridor of our beloved Department of Biochemistry, Maastricht finest. Anxhela, 4 years we spent together and 4 years were gone in a second. I told you in the beginning, and I think I was right. You came to our group as a young researcher, immature, sometimes doubting about research and results, and even sometimes doubting about yourself. In our years together, I've seen you growing from this to a very independent researcher, capable of designing own experiments and high-end research, which we have seen today. You mastered the iPSC work. You mastered the CRISPR work to generate your PAR knockout cells. You did your secondment for almost 1 year in Mainz to isolate smooth muscle cells from mice and isolate extracellular vesicles, and then went to Boehringer in Biberach, somewhere in the middle of nowhere, to do 3 months on microRNA analysis in these vesicles. One can say that you find independence, maturity, and wisdom by traveling, which you did, and you found more. You even found your partner Kostas in Mainz. You're number one, but also ESR number one. But to be honest, we were very happy that you came back to Maastricht. And I can speak on behalf of many people of our group to have you back. Not that you were fully integrated in the Dutch culture, because as I heard, you only know one word in Dutch. Can you say it?
Gezellig.
Gezellig. So actually we would have expected a little bit more, that we have some more words. But we can practice on that. But your enthusiasm and energy are a blessing and a great contribution to our group. And of course, your delicious tiramisu. Although you seem to make it these days without alcohol, as you live since over 2 years a life fully free of alcohol. But from energy at the parties, one can tell that you don't need alcohol to have fun and to be social. Yet, you promised me to raise the glass today. Anxhela, I think you truly found love. And I mean not in Kostas, but in science. For sure, I know that you will make it as a successful scientist. Unfortunately for us, your love for Kostas made you move to Germany, whereas we would have loved to have you stayed in Maastricht. You found a great job in Frankfurt, in the lab of Ingrid Fleming. And one can say this is a top-notch research institute. And also that is solely based on your skills and your growth that you have made in the last 4 or 5 years. Anxhela, coming to an end, I wish you all the best for the future. And I'm pretty sure that you will succeed in whatever you're going to do. For now, the defense is over, the stress is gone. You can start relaxing, whatever that means for you as an Italian. And enjoy and celebrate this fantastic achievement and moment. Dear Anxhela, I hope, but I'm also pretty sure we will stay in touch. Arrivederci.
Thank you.
Dear Dr. Habibi, I also want to congratulate you with this achievement, and I can do that not only on behalf of myself, but also the whole community of Maastricht University. Congratulations. After all these kind words by Professor Schurgers, your promoter, there's not much to add for me. I wish you everything, but especially a lot of success in your future career. Before closing this ceremony, I want to give the floor to Professor Schmidt for a short words.
Yes, there's not much to add to the words of Leon Schurgers. Congratulations, an excellent thesis, and I'm delighted that you continue your scientific career. Everything else would have been a big pity. Congratulations.
Thank you.
Thank you. Professor Reinhardt.
Dear Anxhela, I congratulate you to your thesis, and I think it was a long and maybe also a stressful and labelsome period of your life. But I think you really have shown that you are well equipped for the next steps in science, and I wish you luck with that steps.
Thank you.
Professor Kauschke.
Thank you for letting me participate. Anxhela, big congratulations on your final chapter of your PhD thesis. I think it's a great closure. We could really clearly appreciate your scientific curiosity also during your visit at Boehringer. I would not consider ourselves in the middle of nowhere, but it's great to see hear about your continuation in Frankfurt. Also, congratulations to that. And so looking forward to hearing your footprint, seeing your footprint in the scientific community in the years to come. Thank you.
Thank you.
Thank you. After using this hammer, I invite all the people in the audience to go to the rafter for the reception and to prepare for the reception. We stay a little bit longer in this room to make a few pictures and also pictures on the stairs. And there Hereby I close this academic ceremony.
Congratulations.